So, good evening, students. So, today we'll start with the week 8 uh, assignment session for the last year. So, the topic for the day are techniques to study protein and protein 3, protein interactions and microarrays, protein interactions and systems biology, informatics 2, ethics in research and publications. So, myself, Ankit Kumar Malik, an NPTEL PMRF fellow from IIT BHU, and the course instructor for the same is Professor Sanjeeva Shrivastava from IIT Bombay. So we'll start from the question one. So the question one states that the number of Indian women prone to cervical cancer has leaped over the years. So you wish to create awareness through scientific endeavors, and you have recruited a cohort of 50 females to screen for markers indicating the risk of cervical cancer. So which is the strategy you would like to follow? So some options given here are you may perform fluorescence in situ hybridization on the samples, or you may screen the serum samples using protein microarray for autoantibodies, and you may perform a PCR from the blood samples and isolate proteins. Like both A and C can be done. So we'll start from the some basic concept of fluorescent in situ hybridization. So actually the FISH is a technique used to detect and localize the presence or absence of specific DNA sequences on chromosomes. So in the context of cervical cancer, FISH can be utilized to detect genetic abnormalities such as amplifications or deletion of specific genes associated with the development of cervical cancer. So the technique provides valuable information about genetics alteration that may contribute to the risk of cervical cancer. And here the option B, which is you may screen the serum samples using protein microarrays for autoantibodies. So here the protein microarrays are powerful tools for analyzing the expression levels of multiple proteins simultaneously. And by screening serum samples for autoantibodies, which are antibodies produced by the immune system against body's own proteins, we can identify potential biomarkers associated with cervical cancer. So elevated levels of specific autoantibodies may indicate an immune response against proteins that are dysregulated in cervical cancer cells. And the option C here, you may perform a PCR from the blood samples and isolate proteins. So while performing PCR is a molecular biology technique used to amplify specific DNA sequences. In the context of cervical cancer, PCR can be used to detect the presence of viral DNA, such as human HPV DNA, which is strongly associated with the development of cervical cancer. Isolating proteins from blood samples can also provide valuable information about proteins biomarkers associated with cervical cancer. So, like coming back to the question and options. So, I told you in the serum samples using protein microarrays for autoantibodies. So here that is the option B. So the correct answer for the question one is the option B. So we may screen the serum samples using protein microarrays for autoantibodies. So moving to the next question, that is question 2. So here one diagram is given here. So the diagram describes a type of ionization source in a mass spectrometer. So pick the correct statement. So the options given below here are the diagram illustrates electrospray ionization where the analyte is a gaseous chemical. So the option B is in the illustration of chemical ionization. The analyte is a volatile material. The option C is the process of thermospray ionization is shown in the diagram in which heated particles bombarded the solvent and cause it to ionize. And the option D is the diagram illustrates the Moldy principle, which calls for uniformly mixing the sample with the substantial amount of matrix. So here, Moldy is an ionization technique that uses a laser energy absorbing matrix to create ions. Wait, I'll show you the diagram first. It creates ions from large molecules with minimal fragmentation. So it has been applied to the analysis of biomolecules like DNA, protein, peptides and carbohydrates kind of things, and various organic molecules also, like polymers, dendrimers, and other macromolecules. So which tends to be fragile fragment when ionized by more conventional ionization methods. So it is similar in character to electrospray ionization in that both techniques are relatively soft, uh, like low fragmentation ways of obtaining ions of large molecules in the gas phase. Through MALDI typically produces 4 fewer multi-charged ions. And in MALDI methodology is a three-step process. So the first one is the sample is mixed with a suitable matrix material and applied to a metal plate. Second, a pulsed laser irradiates the sample, triggering ablation and desorption of the sample and matrix material. And finally, the analyte molecules were ionized by being protonated or either deprotonated in the hot form of ablated gases, and then they can be accelerated into whichever mass spectrometer is used to analyze them. So according to this, coming back to the options given here, so the option D, the diagram illustrates the MALDI principle, which calls for uniformly mixing the samples with a substantial amount of matrix. So the correct answer for the question 2 is the option D. Moving to the next question, that is question. So here some match of the following is given here. So match the left column with the right column, and the in the left column the options are gas phase ionization, solution phase ionization, solid phase ionization, and the right column the options given here are electrospray ionization, chemical ionization, photoionization, MALDI, and fast atom bombardment. So I'll cover one by one, then we'll look which are the matching and correlatively things with each other in the left column and the right column. So the first term here is electrospray ionization. So it is a technique. So I'll start with the gas phase ionization first. So gas phase ionization refers to the process of converting neutral gas molecules or atoms into ions by gaining or losing electrons. So the phenomenon commonly occurs in environments where gases are present, such as the Earth's atmosphere or in laboratory conditions within a vacuum chamber. So gas phase ionization can happen through various mechanisms, including electron impact ionization, chemical ionization, or photoionization. And there are three, like electron impact ionization, chemical ionization, photoionization. And the second option given here is electrospray ionization. Uh, so electrospray ionization is a technique used in mass spectrometry to produce ions using an electrospray in which a high voltage is applied to a liquid to create an aerosol. It is especially useful in producing ions from macromolecules because it overcomes the propensity of these molecules to fragment with when ionized. And the other option here is solution phase ionization. And solution phase ionization occurs in liquid solution when neutral molecules or compounds undergo ionization to form ions. Wait, I'll show you. So this process is crucial in various chemical and biochemical reactions occurring in aqueous solutions. So solution phase ionization commonly involves the dissolution of a substance in a solvent where it can either donate or accept the protons or to form cations, or anions respectively. And the last option in the table is solid phase ionization. So the solid phase ionization involves the generation of ions within solid materials. So this phenomenon is observed in various techniques. Hmm. And so in here the semiconductor devices such as transistors and diodes, like solid state ionization occurs when impurities or dopants introduce additional charges carrier, like either electron or some holes, so into the crystal lattice, thereby altering its electrical conductivity. And in solid state ionization is a fundamental operations for secondary ion mass spectrometry and laser ablation inductively coupled plasma mass spectrometry. So now we'll correlate the given options in the left and the right column. So the gas phase ionization is related to the chemical ionization, that is option 2. Like, do we have any like option when where A is relating to the second one, and the option A and C. Then we have solution phase ionization. It is related to electrospray ionization. So B is going to the one. So according to this, the option C is the correct one, where gas phase ionization is matching with the chemical ionization, and solution phase ionization matching with electrospray ionization, and solid phase ionization is matching to the moldy one. Okay, so the correct answer for the question 3 is the option C. So moving to the next question, that is question 4. So here in labeled mass spectrometer experiments, the isobaric tag is composed of two different chemical groups which have specific properties. So select the correct combination of chemicals in the right order from the below mentioned diagram. So here we have the options like here in one diagram is given, one chemical structure is given here. It divided into two parts. That is, one is A and another one is B. So we have to like we have to get it what A is and what B is. So we have to correctly find out the correct combination of chemicals in right order. So the given options and given combinations below here are, so the A is reporter group that provides signature ions in mass, and the B is balance group having a mass of 31 to 28 Dalton, and the option B is causes neutral loss in MS, and balance group maintains charged state and ionization efficiency of a peptide. The option C is the combined molecular weight of A and B always varies sample to sample. The option D is where A states that it's a balanced group gives sufficient B and Y ions. So where B reporter group causing neutral loss of ions. So before getting deep into it, I'll just summarize some of the basic concept and basic theories of the same. So in question one term was that like isobaric tag. So we'll look into what isobaric is. Actually, isobaric refers to molecules that have the same mass but different chemical compositions. So in the context of isobaric tags in mass spectrometry, so these tags are designed to have identical over mass but differ in their chemical compositions. So this property is crucial because it allows for the labeling of different samples while maintaining consistent mass characteristics, facilitating their simultaneous detection and quantification within a single mass spectrometer experiment. So now two different chemical groups was given in the question. So one is reporter ions, and what another was is reporter group and balance group. So we have to look into it what reporter group is and what balance group is. So the option A, this reporter group. So these are the components responsible for generating distinct signals in the mass spectrum corresponding to each labeled sample. So where in the balancing group, so balancing groups are chemical moieties attached to the isobaric tags to ensure that the overall mass of the tags remains constant across different samples. So despite the variations in the chemical structures of these balancing groups, so their mass contributions are carefully designed to offset each other, resulting in an identical total masses for the isobaric tags. So this balanced mass distribution is essential for accurate quantification and comparison of protein expression levels between the two samples or more than two samples. So according to this, the given options here is the A part is the reporter group provides signature ions in MS by MS, and the B is the balance group having a mass of 31 to 28 Dalton. So the correct answer for the question 4 is the option A. So moving to the next question, that is question 5. So in question 5, IIT Bombay, a student from IIT Bombay is performing a protein microarray experiment to investigate protein interactors. So to detect two distinct types of samples against a printed protein, you have tagged two different colored probes within samples. So as a result, you get a third color refers to the image like color 1, 2 and 3 are given here. So which is definitely not a blank one. So given only two types of samples to test interaction with, explain what is the most likely the case. So like the four possible given options are given here. So the option one is the blending of two samples resulted in the creation of a new sample type. The option B is that protein had no effect on any of the samples. And the option C is that protein elicited responses in both samples and overall no differential expression. And the option D is both A and C are true. So like before going into it, I'll tell you some of the basic concept like analytical protein microarray things. So according to the question, so protein chips are made in such the same way as a DNA microarray. So a glass or a plastic surface is supported within an array of molecules designed to capture specific proteins. So these capturing molecules can be other proteins such as antibodies, antigens, enzymes, or peptides, or they can be snippets of DNA or small organic ligands. So in the question, the experimental setup, the student has labeled two different types of samples with distinct colored probes. So these probes are then used to interact with the protein printed on the microarray slide. And the expected outcomes were given that only two types of samples are tested against the printed proteins. So we anticipate observing two distinct colors corresponding to the labeled probes used for each sample type. So the unexpected third color in here in the question they are saying that you get a third color. So this is the unexpected third color. So we'll look the reason for the unexpected one. So the emergence of a third color on the microarray distinct from the colors of the two labeled probes and the blank. So which suggests an unexpected phenomenon. So this third color could potentially indicate a non-specific interaction or a cross reactivity between one of the sample types and a protein on the microarray. Okay, so I'll give you some like possible explanations for the same. So there are some three types called non-specific binding and the cross reactivity experimental artifact and some further investigations. So the non-specific binding, so it's possible that one of the sample types or its associated probe is exhibiting non-specific binding to proteins on the microarray, and it could be the cross reactivity. Like alternatively, there might be some degree of cross reactivity between one of the sample types and a protein that was not the intended target. And the last one is the experimental artifact. So it's also essential to consider the possibility of experimental artifacts such as issues with the probe labeling, sample preparation, or microarray processing, which could contribute to the observed results. Okay, so now we'll look to the possible options given here. So according to this, the option C, which states that the protein elicited responses in both samples and overall no differential expression. Okay, so the right answer for the question fifth is the option C, that says that the protein elicited responses in both samples and overall no differential expression. Okay, so moving to the next question, that is the question 6. So here we have some three diagrams over here. So please select the protein microarray type from the below diagram in the correct manner. So here we have three options like direct immunoassay, reverse phase immunoassay, or sandwich immunoassay. So we have to detect or look out which is the direct one, which is the reverse phase, and which one is the sandwich immunoassay. So we'll cover one by one. Like I'll explain the direct, reverse phase, and sandwich one by one with their mechanism, with their application, and with their detection. So first we'll cover the direct immunoassay. Okay, so in direct immunoassay, so the mechanism of the direct immunoassay is in a direct immunoassay, the target analyte, which could be protein or any antigen, is directly labeled with a detectable marker, like detectable markers like fluorescent or enzymatic label. And the application for the same is it is suitable for detecting small molecules, peptides, or proteins with high specificity. And the detection for the direct immunoassay is the signal is directly proportional to the concentration of the target analyte, allowing for quantitative analysis. And the second one is reverse phase immunoassay. So here the mechanism for the reverse phase immunoassay is that the target analyte is immobilized on a solid surface and labeled antibodies are used for detection. So the labeled antibodies bind specifically to the immobilized target analyte. And the application for the reverse phase one is reverse phase immunoassay are employed when the target analyte is abundant and can be effectively captured on a solid surface. Okay, and the detection for the reverse phase immunoassay is that the signal. Okay, we'll just wait for a minute. I'll be back. Hanji. So the third immunoassay is the sandwich immunoassay. So in a sandwich immunoassay, the target analysis sandwiched between two antibodies. One is immobilized on a solid surface, and the another labeled with a detectable marker with their detection antibody. And the application for sandwich immunoassay are particularly useful for detecting larger proteins or molecules where multiple epitopes are available for antibody bindings. Okay, so according to all the definitions and their applications and mechanisms and detection, we'll look for the same. So according to this, the first one is looking like the direct immunoassay. So in where how can we detect that it is a direct immunoassay, not sandwich or reverse phase? So here the target analyte is directly labeled with the detectable marker. Okay. So the second one is the sandwich immunoassay. So as you can see, here the target analyte is sandwiched between two antibodies, one immobilized on a solid surface and other labeled with the detectable marker. And the third one is reverse phase immunoassay. This third one is reverse phase. Okay. So here, as you can see, the target analyzed is immobilized on a solid surface, and label antibodies are used for detections. Okay, the labeled antibodies bind specifically to the immobilized target analyte. So the correct answer for the question 6 is the option B. The first one is direct immunoassay, second one is sandwich immunoassay, the third one is reverse phase immunoassay. Okay, so moving to the next question, that is question 7. So DAPA, DNA array to protein array. DAPA was invented to overcome a few hurdles of conventional protein array technologies. Which of the following was the biggest hurdle of all? So the options given here are the high density printing of conventional protein microarrays was not possible, and the option B is unavailability of purified functional proteins for immobilization on arrays, unavailability of DNA sequences from which transcription and translation could be performed, protein-protein interactions could not be found using conventional protein microarrays. So here, so there's one thing is called DNA array to protein microarray, which represent an innovative approach to protein array technologies, where DNA sequences encoding proteins of interest are directly printed onto the array, allowing for the subsequent in situ transcription and translation of these sequences to generate functional proteins. So this method offers several advantages over conventional protein arrays technologies. So now let's examine the options to identify the most significant hurdle addressed by DAPA. So the options, according to the options given here are, the option first is the high density printing of conventional protein microarrays was not possible. So while high density printing could be a challenge in a conventional protein microarray, it's not the most significant hurdle addressed by DAPA. So high density printing limitations can be overcome through advances in printing technologies and array fabrication methods. Okay. The option B here is the unavailability of purified functional proteins for immobilization on array. So here, so this hurdle is indeed significant in conventional protein arrays technologies. So obtaining purified and functional proteins for immobilization onto arrays can be challenging and time consuming, limiting the utility of these arrays for high throughput applications. Okay, and the option C, unavailability of DNA sequences from which transcription and translation could be performed. So here the option highlights a key aspect addressed by the DAPA. So in conventional protein microarrays technologies, so obtaining DNA sequences and coding proteins of interest can be a bottleneck. So DAPA circumvents these limitations by directly printing DNA sequences onto the array, allowing on the on-site synthesis of proteins via transcription and translation. And the last option here is protein-protein interaction. So while this is a challenge in conventional protein microarray, it's not the most significant hurdle addressed by DAPA. So protein-protein interaction studies can be performed while like using approaches. The conventional protein microarrays can be still be a valuable tool in this regard. Okay, so according to this, the right answer for the biggest hurdle of all in the DAPA is the option B, like the unavailability of purified functional proteins for immobilization on array. Okay, so the right answer for the question 7 is the option B. So like moving to the next question, that is question 8. So the protein LDHA is one of the variables differentially expressed in the HPV positive and HPV negative human cell lines. So using bioinformatics, we want to identify LDHAs in silico protein interactions. So what software or web server do you wish to employ as a result? So select the appropriate combinations from the list below. So here we have some core software, or you can say web server, as given here are MaxQuant, String, Cytoscape and PyMol. So according to this, I'll show you some of the interface of Cytoscape and String. The correct answer for this question is the Cytoscape and String. So these two softwares are used for the to identify in silico protein interactions. So we'll look into the interface for the both of the softwares now. So first we'll cover with the Cytoscape. Okay. So this is the interface of a Cytoscape, and this is the network created by the software called Cytoscape. The Cytoscape you can use either by like as a software, or you can use as a web server also. So this kind of network can be prepared by, this is the network of by String, and this is the network by Cytoscape. So now I'll clear some of the concepts what Cytoscape and what String is. So Cytoscape. So Cytoscape is a powerful software platform for visualizing and analyzing complex networks. So including protein-protein interaction networks generated from data obtained through string or other sources you can say. So after obtaining the list of potential LDHA interactors from string, so Cytoscape can be used to visualize the interaction network platform, further analysis like network topology analysis, identification of functional molecules, and gain insights into the biological significance of the identified interactors. And the next one is the string. And string is a widely used database and web resource for predicting protein-protein interactions. So it integrates both experimental data and computational predictions to generate comprehensive interaction networks. So by inputting LDHA as the query protein, so along with the information about the cell lines being studied, so Sting can provide a list of potential protein interactors based on various types of evidences like such as experimental studies, text mining and computational predictions. So okay, so the correct answer for the question 8 is the C, where it states the string and cytoscape is the appropriate combinations to employ as a result over here. Okay, so moving to the next question, question 9. So here we have one crystal structure given in the question. So identify the crystal structure for like CW 6W 9C from the diagram. So what is the resolution of the crystal structure? So pick the correct combination. So and for this question, we do not have to do anything, and we do not have to do some like basic things. It's just a kind of Google search. So we just have to like copy this crystal structure name like 6W9C, and we'll paste this in the PDB website called Protein Data Bank, and then we'll code the results like this kind of result we'll get. So there you can get the crystal structure for the same code and the resolution for the same code. So resolution here given here is 2.70 angstrom, and the diagram you can see easily. So now what are the possible given options here are? So here in option A it is crystal structure 1 and resolution is 2.70 angstrom. Okay, so according to this the correct answer for the question 9 is option A. Okay, so moving to the last question, that is question 10. So the question 10 states that the component of a mass spectrometer that separates ionized masses depending on charge to mass ratio is called a mass analyzer. So pick the one which is not used as a mass analyzer. So the given options here are magnetic sector, fast atom bombardment, quadrupole, and a time of flight. So I'll explain one by one what magnetic sector is. So magnetic sector here is like a magnetic sector mass analyzer separates ions based on their mass to charge ratio like m by z. So by passing them through a magnetic field, so the degree of deflection of the ions depends on their mass to charge ratio, allowing for separation and detection. And magnetic sector mass analyzers are commonly used in magnetic sector mass spectrometers. Okay, and this other option is like fast atom bombardment. So the fast atom bombardment is a method of ionization rather than a mass analyzer. Fab involves, in layman language, we call this as a fab. That is fast atom bombardment. So fab involves bombarding a solid or a liquid sample with high energy atoms, usually xenon or argon, and to produce gas phase ions for analysis in a mass spectrometer. So it is not used as a mass analyzer but rather as an ionization technique. Okay, and the other options is time of flight and quadrupole. So time of flight, as you can all you know, on a time of flight mass analyzer, ions are accelerated through a vacuum tube by an electric field. So the time it takes for ions to travel a fixed distance depends on their mass to charge ratio. Lighter ions reach the detector faster than the heavier ones. Okay, so allowing for separation based on their mass. So time of flight mass analyzers are utilized in time of flight mass spectrometers. And the last given options is quadrupole. So quadrupole mass analyzers uses radio frequencies basically and direct current also. Electric fields to selectively transmit ions based on their mass to charge ratio. So the quadrupole consists of four parallel metal rods and that an oscillating create an oscillating electric field, allowing only ions with specific m by z ratio to pass through the detectors. Okay, so according to this, the given options here are, so the correct answer for the same is the fast atom bombardment, because the other options cannot be used as a mass analyzer but rather than as an ionization technique. So this option is not used as a mass analyzer, while this magnetic sector, quadrupole, and time of flight can be used as a mass analyzer. So the correct answer for the question 10 is option B, that is fast atom bombardment. So this all for today. So thanks students. So you guys can leave the session. So we'll meet in tomorrow's and extra session. So thank you. You guys can leave the session.